Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are delighted to be joined by our guest, Dr. Sara Amalie Solheim. Listeners, Sara is a former PCC Fellow, and she is currently a researcher and Athlete Passport Management Unit, or APMU manager, at the Norwegian Doping Control Laboratory and Nordic APMU. And today, listeners, we're excited to discuss Sara's career path, her work at the Norwegian Doping Control Lab, and her PCC-funded research project. So, Sara, welcome to the show today. How are you?
Oh, thank you, Marie. I'm good, thanks. We have a sunny day in Oslo.
Oh, wonderful. I'm jealous. We've got a cloudy day here in St. Louis. Well, it's great to have you here with us. And for our listeners who haven't met you yet, can you perhaps start by telling us more about yourself?
Thank you very much. I am a 34-year-old human physiologist and anti-doping scientist from a city called Halden, a bit south of Oslo in Norway. And I work as a researcher and APMU manager here at the Norwegian Doping Control Lab and the Nordic APMU. And on the personal side, I love spending time with my family and friends and to be active. So when I'm not at work, you can often find me gravel riding or cross-country skiing in the fantastic forest surrounding Oslo. And I'm also quite a coffee geek and sourdough baking enthusiast. So I love a good Hamburg coffee and a sourdough bread or bun.
Well, Sara, that all sounds absolutely amazing. So can you share with us how you first became interested in science and this field of anti-doping? I think everybody's got their own story of how they've discovered this niche field.
Well, I've always been really interested in sports, and I practiced multiple sports in my childhood. And inspired by my older brother, I decided to apply to a sports and physical education program in upper secondary school. I don't know if every country has that. I guess they don't, but at least in Norway, that's an opportunity. And while studying or during that program, that further sparked my interest in sports science. So I ended up taking a bachelor's degree in sports biology at Norwegian School of Sports Science. And then on top of that, a master in human physiology at the Copenhagen University. So yeah, here I was interested in individual differences in human physiology and the understanding of human exercise performance and its limitations. And since fairness in sports always has been very important to me, both when practicing sports myself and when watching as a spectator, I was also very interested in how these limitations of human performance can be manipulated. So doping. Then I started looking into different opportunities for my master thesis within this field. And I was fortunate to join the group of Professor Nikolai Norsborg and specialized them in autologous blood transfusion during my master's thesis. And then, if I recall correctly, in parallel there, I started working as a doping control officer at Anti-Doping Denmark. And that was my way into anti-doping science and anti-doping work. So I've just been more and more interested in the field after that.
I think that's awesome. And it's cool to hear how you took advantage of these opportunities as they arose. And you mentioned that you were interested in a variety of different sports growing up. What sports did you play?
As many kids, I tried many different sports: handball, soccer, running, orienteering, ballet, etc. But mainly then soccer. And then after that, running that I practiced. And at some point, I started focusing only on running and trying to become as good as I could at that time. So I loved doing sports.
That's awesome. And you mentioned that you were a doping control officer for a bit as well. What was that experience like?
It was a really great opportunity to see more of that side of anti-doping work and how samples are collected and to oversee that everything is done according to the WADA guidelines, and also to see this different sports up close. It was a great experience, and I would recommend, if people have the opportunity to work as a doping control officer, which is often a part-time position, I would recommend doing so because you get to see sports from a different perspective.
Definitely. And I'd love to hear about how the PCC fits into this story. So can you share, Sara, how you found out about the Partnership for Clean Competition, maybe in general, as well as the PCC Fellowship specifically, and how you decided to pursue this opportunity?
Actually, my PhD was also partly funded by the PCC, So I've known about the organization for quite some time, and I also knew people who had been PCC Fellows. So that's how I heard about it. And I think it was back in 2022, in March or April, we submitted an application for the fellowship. And at that time, I worked in a temporary position as a scientific manager at Anti-Doping Denmark, where I worked with research and development work, and also the evaluation of the athlete biological passport. And in this position, I was part of a Nordic collaboration called Nordic Science and Testing Working Group, which is a collaboration between 5 Nordic anti-doping organizations like Norway, Denmark, Sweden, Finland, and Iceland. I don't recall at what point, but that's one of the meetings we started discussing how we could collaborate and conduct some applied research projects together, and then where we could apply for funding. And since several of the group members had some previous experience with successful PCC grants applications, me included, we decided to go for one of those. And also since my position then in Anti-Doping Denmark would finish in June that year, and I wanted to move back to Norway, then we thought that maybe we could combine it with a PCC fellowship for me at the Norwegian Doping Control Lab, and actually together with the Norwegian School of Sports Science, which is well known within exercise physiology. So then we would have expertise within anti-doping, like doping analysis and also exercise physiology. So then we decided to pursue this fellowship and apply for a research grant in parallel to cover some of the research ideas we had in this group.
Excellent. And then, of course, all of the pieces fell into place. You were successful, particularly in this fellowship application, and your PCC-funded fellowship focused on further developing blood testing with a special focus on the athlete biological passport, or ABP. So, Sara, can you talk a little bit more about how you chose this particular topic for your project and why it's so important in the field of anti-doping?
As I mentioned, this application was initiated by this Nordic collaboration between NATOs and the APMU. So we really wanted to focus on a topic that is relevant and can solve practical challenges in the everyday life for as an anti-doping organization and as an APMU. So originally we planned on a 3-year project focusing on, as you said, the ABP and deterrence virtual testing, and also some standardization procedures related to sample collection. But unfortunately, that grant was not accepted for funding. So we had to go through a couple of more rounds to get funding. And then we also got the opportunity to redefine the scope. And after some rounds, we ended up focusing on non-analytical factors that may influence the ABP. More specifically, that was standardization procedures related to cool shipment of blood samples, because they have to shipped cooled for the hematological module. And then also evaluation of some confounding factors like low energy availability, resistant training, and menstrual cycle for the steroid and endocrine profiles in serum. Because this was, as I said, back in 2022, and the serum steroid profile and endocrine profiles or modules, they would be introduced then in 2023. So that's why we ended up looking at some confounding factors we found relevant back then for the passport. Maybe we can talk more about that later. I know that you've had guests before, like Reid and Brad from WADA. They have described in detail what the ABP work is, so I don't have to go into detail about that. But knowledge about how other factors than doping affect the ABP is of great importance. So that's why we wanted to focus on that. And hopefully the work, when published, will be of importance for anti-doping organizations, APMU managers like myself, and experts when they interpret and evaluate these passports.
Absolutely. And I think the ABP is complex. I think there are so many different confounding factors and things that we're still learning about the ABP that there's a lot of unanswered questions. So it can be difficult to narrow it down. So Sara, what were the specific goals or aims of your PCC Fellowship research project?
It can be difficult to narrow it down, as you said, but we wanted to focus on the evaluation of non-analytical factors that may influence the ABP and that could be relevant to our work in the NADOs and the APMU. And one potential confounding factor we wanted more information about is low energy availability, LEA in short. And according to the IOC consensus statement, LEA is any mismatch between dietary energy intake and energy expanded in exercise that leaves the body's total energy needs unmet. And LEA is frequently reported in sports with high training volumes, especially in leanest demanding sports. And we humans, we're adapted to cope with periods of LEA by downregulating biological processes that are temporarily unnecessary, such as, for example, the reproductive system and bone production. And research has shown that LEA can lead to suppressed anabolic hormones and reduced bone formation markers such as IGF-1. And knowing that some of these markers are included in the serum steroid module and endocrine module, we wanted to evaluate if there are differences in biomarkers in the steroid and endocrine profiles between male and female athletes with LEA versus male and female athletes without symptoms of LEA. And this would help us interpret the passports where we think that LEA could be a contributing factor to what we're seeing. So this study was part of a larger study conducted at the Linnaeus University in Sweden with Professor Sara Amalie as principal investigator. This study was also partly funded by WADA. So that was one of 3 projects we had. And another potential confounding factor we wanted to evaluate was acute exercise and also menstrual cycle in women. So it's well known how acute resistance exercise increases serum testosterone levels in healthy men, but it's less clear how it affects testosterone in women. And as for many things, there are more studies done on men, right? So that also comes for, of course, menstrual cycle. We have some information, but not all studies have been well designed, and thus the picture is not fully clear. And therefore, we wanted to evaluate how a standardized resistance training session affects the steroid and endocrine profiles in female weightlifters and powerlifters in 2 different phases of the menstrual cycle. A secondary aim of this project was to evaluate if the recovery after the training session varies throughout the menstrual cycle. And this intervention was conducted at the Norwegian School of Sports Sciences and the analysis at the Norwegian Doping Control Lab. So we had a great setup there with exercise physiology expertise and doping control analysis expertise. And the study was partly funded by WADA. So as a physiologist at base, I am also interested in other factors or areas than anti-doping. So it was very cool for me to be able to add this other touch and different aspect of this study and to hopefully contribute to more knowledge about the female athletes. And actually we had one more project. In this 3rd and last project we started during my fellowship, we looked into one practical challenge related to ABP blood sampling, which is strict transportation conditions. So all samples collected for the hematological module of the ABP must be transported refrigerated to the lab for analysis. But when the results are entered into the ABP management system called ADAMS, samples are per default deemed invalid if the temperature logger accompanying the samples have registered temperatures below 0 degrees Celsius, meaning that, it has been really cold for the samples. So for the anti-doping organization, this means that samples might not be used in the passports, and therefore it's a waste of resources and also kind of an ethical consideration as well for the athletes who have provided the samples. So therefore, we wanted to evaluate what happens to the ABP parameters when exposed to cold transport conditions, and also for how long time and ABP blood samples can be kept at relevant sub-0 degrees Celsius without the markers in question being affected. So the running costs for this study were covered by the 4 involved Nordic anti-doping organizations and the Nordic APMU. So those were the 3 projects we initiated during my fellowship. 3 quite different goals, but all of them were some kind of under non-analytical factors for the ABP.
Absolutely. And I think these are really exciting and important questions, these 3 that you enumerated. And I love the point that you made about bringing your exercise physiology interests and expertise to the table, because I think that is one of the things that really makes anti-doping so exciting, is that it is this interdisciplinary field with people coming in from different backgrounds. And I'd love to talk a little bit more about these projects. Can you share an update on the progress or the status of each one of these 3 projects?
Yes, for sure. I recently came back to work after maternity leave, so I'm trying to catch up on things. But luckily, I have a really good research team, or teams actually, for these several projects. And they continued the intervention and data collection for the project about resistant training and menstrual cycle when I was at maternity leave. And now we're in the process of finishing that data collection. And hopefully soon we will end the collection and start looking into the data. Unfortunately, we have had some difficulties of the participants dropping out due to the overall load of being a participant and combining with their everyday work life or being a student. That's why it's taking very long. Also, working with the menstrual cycle, there are many things that could influence the cycle so that the participants lose their menstrual cycle and then can't participate anymore. So it's a very good experience, actually, to work with projects testing based on the menstrual cycle. And for the 2 other studies that I mentioned previously, we have finished the data collection. But again, I'm running the 3 projects. So when one is taking up the whole time of sample collection, then the 2 others have to be on hold.
Mm-hmm.
So although we have completed the sample collection, we have not completed the analysis of all the data and all the material. We will start doing that soon as well. And I'm very much looking forward to this next step. And also to look at what the findings and write up some manuscripts, and then hopefully see the results being implemented as we hoped by anti-doping organizations and the APMUs. So the status now is hopefully to move forward to the sample analysis and data analysis part and wrap up the studies, and hopefully within not that long time also publish the results.
Well, Sara, it sounds like you have a lot of exciting things on the horizon, which is wonderful, and perhaps a lot of lessons learned in these studies. I think research takes time, and there are some major challenges, and I think a lot of people don't necessarily realize it. It feels like science is just sort of a recipe where you follow the steps and you get results and everything works great, and so many of the times that's not the case.
For sure. It's a lot of up and down and back and forth, and that's how we're learning, and can take on some lessons when designing upcoming projects.
Definitely. And it sounds like these projects will be wrapping up relatively soon. So thinking about the future, what are some key challenges or barriers that remain when it comes to the ABP that you think future research should address?
Well, I would start with saying that the ABP is a great tool, but it's complex, and there are still many things that we're learning about the ABP, and it's still possible to further develop this tool. And as I see it, one challenge lies in the nature of the ABP. It's an indirect method where we are trying to detect the effects of a doping substance or a method rather than the substance itself directly. And these biomarkers included are naturally present in the body. So for correct interpretation of the passports, it's critical to consider how other factors or external elements can influence the values of a given biomarker. We have talked a lot about some confounding factors earlier in this podcast, and luckily we have valuable knowledge and literature about many factors affecting the included biomarkers. However, in my daily work as an APMU manager, I realized different research projects that could emerge from this work, and I think that it's always good with more information about potential confounding factors that could help our interpretation of the passports. I would also say that another thing that could be useful is to look further into new biomarkers that could be included in the ABP, or also other ways to use existing data to help the ABP even more sensitive and specific through, for example, machine learning. For example, of biomarkers, I see a huge potential in including iron metabolism indicators and also indicators of plasma volume change, which I know some research groups are already looking into.
Well, Sara, I appreciate you sharing more about your PCC-funded research, and I'd love to talk kind of more broadly about the PCC Fellowship experience. So can you tell us what it was like to be a PCC Fellow?
I really liked it. I don't know if it's still like that, but back then we could choose or apply for one of two different tracks, like a lab track and more an academic track. We kind of ended up doing a mix of those. Like I had a joint fellowship position at the Norwegian Doping Control Lab and the Norwegian School of Sports Science. And this collaboration between a WADA accredited lab and what I would say is a world-class exercise physiology research environment was really great. And then on top of that, we had this anti-doping organization within the mix of the collab, which was a really, really good setup for me with the knowledge of each members involved and also the facilities and the mentorship. Because as a fellow, you have to choose a mentor. And for me, then with two institutions, I had two mentors. And that was a really nice thing that you're not alone in this journey and you can learn from the best within this field. So I had Yvette Denes, which is the head or director of the WADA accredited Oslo lab. And I also had Professor Klaus Madsen at the Norwegian School of Sports Sciences as my mentor. So that was really great. As I said, this setup was really good. So then I could run the experiments at the Norwegian School of Sports Science, which is a great environment if you're really into sports, and then analyze the samples at the Norwegian Doping Control Lab, which is really good when it comes to doping analysis. So then during my fellowship, I could dive into those two fields. I would also like to point out that within the description of the fellowship, it's not only to focus on the research only, it's also more about the journey of creating good candidates to continue work within anti-doping. And for me, this fellowship provided me with this opportunity to continue to work within the field. So at first, we extended the 2-year fellowship with some months so that I could continue with the research projects that were ongoing. And then after that, the fellowship kind of created an opportunity for me to continue working as the Norwegian Doping Control Lab, and APMU created a new permanent position that I'm currently in now, involving research and APMU work. So I don't think that would have happened if I didn't have this fellowship at the Norwegian Doping Control Lab. It was a really good experience leading into this opportunity that I have now. And I gained a lot of different relevant experiences for my current job. And I'm so happy that I can continue to contribute to the field of anti-doping science.
Absolutely. And as part of your fellowship, you were able to attend the PCC conference as well. So can you talk about your conference experience and what it was like talking about your fellowship work with others there?
Actually, that was not my first time during my fellowship. It was not the first time there. I think it was my third PCC conference.
Mm-hmm.
And in my opinion, it's one of the best and most relevant when it comes to anti-doping research. I really also like the fact that it's not only scientists from anti-doping labs represented at this conference, but also individuals from different sports federations and anti-doping organizations. You have this diverse expertise represented, which I think create a great platform for exchanging ideas and coming up with new research possibilities and collaborations, because it's not only interesting presentations going on, but you get the opportunity to talk to different people in the breaks between the presentations. And that's when some really good research opportunities arise. So yeah, it was really great attending, but also, as you said, to present at the conference was definitely a great experience for me as a young researcher.
Absolutely. And we'll dive into, I guess, what you're doing right now. You mentioned that some of the things that you learned during your fellowship are useful now in your current role. So can you talk about what your current role looks like, or what does an average day or week look like there at the Norwegian Doping Control Laboratory in APMU?
Well, I work, as you said, both as a researcher and APMU manager, like 50-50. This means that actually 50% of the time I'm evaluating profiles from the hematological module of the ABP, athletes from different sports and nationalities. And this involves the assessment of sample validity, which is related to one of the projects that I mentioned earlier. Also then followed by assessing the new values in relation to the athlete's history, and then finally providing recommendations for future testing or additional analysis to the passport custodian, who is the owner of the passport. So that's like what I do 50% of the time. And then the other 50% is dedicated to the running of research experiments, writing of ground applications or manuscripts, and also analyzing samples or trying to further develop analytical methods. And then actually usually a combination of all. So as a researcher, you have to have many things going on at the same time. So an average day is difficult to describe, but it's a mix of all these things. And some days I do only APMU work, and some days only some research project. But I think it's a really good mix. And also, as I touched upon earlier, the APMU work informs research, and then the research also informs ABP work. So my roles are closely connected. So while I work with these profiles, I get inspired to what we can apply for funding for and get more information about as in the other part of my position.
And that makes sense. And Sara, it sounds like a delicate balancing act with many different responsibilities. So we wish you, of course, the best in this exciting role. And I'd love to just talk a little bit more about your experience as an early career anti-doping researcher, because I think this is a challenging period where a lot of people may drop out of the field. So what have been some of the biggest challenges that you've faced in this early part of your career?
You're right. It's being within academia and not only anti-doping. It can be challenging. I think I'm not the only one pointing out that one of the biggest challenges is related to funding, and actually receive funding for research projects and salary, basically, to continue having a job. But luckily for me, I've been surrounded by great experts and mentors at all the institutions I have been affiliated with, and they have taught me a lot about how to write successful grant applications. So that I'm grateful for, but it can be really challenging. And of course, not all application is successful. Another challenge that I want to point out is that there are not that many job opportunities within academia, and most of them are temporary positions. And with that comes a lot of uncertainty. That has also been a big challenge to know if you will have a job in a long time from now or not. So those two are very closely linked, like the funding and job opportunities, but that's, I would say, the biggest challenges.
Absolutely. And I think one of the goals of the PCC with this fellowship opportunity is to help people bridge that early career period and help them land those permanent opportunities. So can you talk about some of the things that you learned during your fellowship that have been useful now that you are in a permanent position?
Well, during my fellowship, I got involved with the APMU. So one of the things, if you're at a lab fellowship, it's like mandatory or to work in the lab. My part of that was to work at the APMU. And also then I got the opportunity to start evaluating passports, and also got the opportunity to enhance my experience within lab work, as I was supervised with some senior scientists and other lab staff in how their routine doping analysis is conducted. So that was very useful. And now as I work as an APMU manager. And another part of that is that the fellowship provided me with the opportunity to take on an increasingly independent role in planning, designing, and conducting research projects, and also the writing of successful grant applications and ethical approvals, which are key tasks as a researcher. I had to do all those things during my fellowship, and I still have to do it. So definitely got some more experience, and which is of great value now.
Certainly. And for listeners out there who might be considering applying for a PCC fellowship with the next funding cycle, can you share any of the other benefits that you've enjoyed, or maybe advice for applicants who are working on their applications?
I would say the first PCC fellowship is a valuable step for anti-doping careers. As you said, the goal is for the young researchers. It's supposed to be a stepping stone into a career within the field. And one of the really good things about this fellowship is you have to have an expert mentorship throughout the fellowship and guidance then for building an anti-doping scientist career. Really nice thing also related to what I said earlier. You have the safety of guaranteed salary, which means that you can focus on running impactful anti-doping experiments in a timely manner. And also fellows, they gain a lot of relevant knowledge, either, or in my case, both from an anti-doping lab or an APMU and work at an academic institution, which can be used to conduct research within anti-doping or, of course, also the research outside of anti-doping. So there are a lot of benefits of conducting a PCC fellowship. So I would definitely talk warmly about it and recommend people to apply.
Fantastic. Well, Sara, we appreciate you sharing your fellowship experience with all of us today. And one of my favorite questions to ask everybody, we touched on just some of the limitations and the challenges that you face, but the fact that there are so many unanswered questions in anti-doping. So if we gave you all of the resources you could dream up, funding was not a problem, staff, technology, feasibility, what is the one problem in anti-doping that you would most want to solve?
I'm not surprised that you're asking this one because I think I've been listening to the podcast and I think you ask this at the end of all episodes, right?
Mm-hmm.
And without listening to other podcasts, I would actually mention autologous blood transfusion also as one thing I would like to see a method being developed for. That's also what my friend and former colleague and mentor Jakob Merkerbeit replied. So it's not that very original answer, but it's based on my background and how I first got into anti-doping research through looking at performance effect of autologous blood transfusion. But as he mentioned in that episode, so now we don't really have a direct detection method for it, but that would really help my daily work as an APMU manager. And I know that several research groups are trying to solve this problem. But if I had to choose something else, if I get to say two things, I think I would like to conduct a large-scale study trying to further develop and evaluate actually virtual testing with capillary blood. During my PhD, I looked a lot into dry blood spots, but I know that also there's a lot of research going on into liquid capillary blood use and the use of that within anti-doping and doping control. And one of the benefits of these collection methods is the potential time and cost efficiency compared with, for example, collection of urine sample, which can take quite some time, especially if the athletes are dehydrated. But I think that the potential of these collection methods can be fully exploited if it was possible to do it virtually with the guidance, of course, of a doping control officer via video. And I know that some pilot studies have already been looking into that. However, if this protocol or such a collection protocol is to be implemented by anti-doping authorities, it's necessary to have a very robust procedure, which on the one hand preserves, of course, the legal rights of the athletes, and then on the other hand, prevents the athletes from tampering with the process or the samples. So I think there's a long way for that to be implemented, or maybe will never be implemented. But it would be really cool to try to develop both the technicalities like the apps or whatever that is needed for this, and also to try it out in a large scale to see if it's feasible.
Absolutely. And Sara, I agree with you that these are two really important and big areas in anti-doping where there are unresolved issues. So to be able to address these areas of detecting autologous blood transfusion and working out just all of the complex logistics of being able to actually do virtual testing, I think would be absolutely amazing. So we appreciate you sharing your insights and thoughts with us. And if our listeners are interested and they want to learn more about you and the work that you're doing, what is the best way for them to get more information?
Well, if they want to learn more about my research, they can definitely find the publications on, for example, PubMed. But if they want to contact me or learn more about my career path, then LinkedIn or ResearchGate maybe, and contact me through LinkedIn is possible.
Perfect. Well, listeners, definitely take some time to learn more about Sara's research and the work that she's doing there, and get connected if you have specific questions. And Sara, it's been such a pleasure to chat with you today. We really appreciate you taking the time to be on this podcast.
Thank you for having me, and I hope it was a bit clear. But yeah, as I said, I just came back from maternity leave, and being a mother and having not the greatest sleep can mess up the brain somehow, but I think I'm handling it quite well being back to work, but it's an interesting situation, I would say.
Well, you're doing an amazing job, Sara. We admire you and the fantastic work you're doing in anti-doping. So it's been great to chat, listeners. Wonderful to have you here with us as well today. Please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and/or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.